Good morning, everyone. My name is Ayush Kumar. I am currently pursuing Bachelor's of Technology in Computer Science and Engineering from Sri Ram Sarup Memorial University. My roll number is 051. Today, I am here to give a presentation on the topic cell cycle. The contents of the presentation are: what is cell cycle, phases of cell cycle, and cytokinesis. Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, cell replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in accordance way to ensure correct division and formation of progeny cells containing intact genomes. This sequence events by which a cell duplicates its genome synthesis the other constituents of the cell and eventually divides into two daughter cells is termed as cell cycle. Although cell growth, also called as cytoplasmic increase, is a continuous process, DNA synthesis occurs only during the specific stage in the cell cycle. The replicated chromosomes DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. This is the visual representation of the phases of cell cycle. The cell cycle consists of two phases. Let's understand about the phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from a cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases: interphase and M phase or mitotic phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is significant to note that in the 24 hours average duration of cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes or karyokinesis, and usually ends with the division of cytoplasm or cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: the G1 phase or gap 1 phase, the S phase or synthesis phase, G2 phase or gap 2 phase. The G1 phase corresponds to the interval between the mitosis and the initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows and does not replicate the DNA. S or synthesis phase makes the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted by 2C, then it increases to 4C. However, there is no increase in the chromosome numbers. If the cell had diploid or 2n number of chromosomes at G1 phase, then after S phase the number of chromosomes remain the same, that is 2n. In animal cells during the S phase, DNA replication begins with the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized by preparation for mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division. Example heart cells and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase and enter an inactive stage called quiescent stage or G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. The animal's mitotic cell division is only seen in the diploid somatic cells. However, here are few exceptions to this. Their haploid cells divide by mitosis. For example, male honeybees. Against this, the plants can show mitotic division in both haploid and diploid cells. The G1 phase. It is the first phase of the interphase which starts at the end of the previous M phase and occurs till the beginning of DNA synthesis. The G1 phase is also called the growth phase as during this phase the biosynthetic activities of the cell is at a high. The cell increases its supply of protein and the number of cell organelles increases. After these activities are completed, a cell has three options. First, it can continue in the cell cycle and enter the S phase. Second, it can stop in the cell cycle and enter the G0 phase for differentiation. Third, it can become arrested in the G1 phase. The deciding point is called the checkpoint or the restriction point. It is regulated G1 by S cyclins, which causes the transition from the G1 phase to the S phase. The G0 phase of cell cycle is the resting phase of the cell cycle, and the cells in the G0 phase of the cell cycle are called resting cells. Cells such as neurons leave the cell cycle to enter this phase and they never divide again. The absence of nutrients or growth factors causes cells to enter the resting phase. The S phase. This is the intermediate phase that takes place between the G1 and the G2 phase and is the third stage in the sequence of cell cycle. It is a highly specialized phase of interphase and the word S stands for synthesis. DNA synthesis takes place in this phase. Before a cell can divide, it must produce a new copy of the chromosome. For making a new copy of the chromosome, it needs both the replication of the long DNA molecule in each chromosome and the assembly of a new set of chromosomal proteins onto the DNA to form chromatin and chromatid. By its end, each chromosome has been copied to two complete chromatids, which remain joined together at their centromeres until the M phase that soon follows. The G2 phase. The period from the end of the S phase until mitosis is called the G2 phase. It is usually the shortest part of interphase. In this phase, intensive cellular synthesis occurs. Mitochondria and chloroplast divide and energy stored increases. Mitotic spindle begins to form in the interphase. There are two control points such as G1 by S and G2 by M at which the cell takes a decision to whether to proceed or not to the next step. Two control points are also called as checkpoints. The M phase. This is the most dramatic period of the cell cycle. involving the major reorganization of virtually all components of the cell, since the number of chromosomes in the parent and progeny cells are same. It is also called as equational division. Though for convenience, mitosis has been divided into four stages of nuclear division or karyokinesis. It is very essential to understand that cell division is a progressive process and very clear cut lines cannot be drawn between the various stages. Karyokinesis involves the following four stages: prophase, metaphase, anaphase, and telophase. This is the visual representation of mitosis. Prophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G1 phase of interphase. In the S and G2 phases, the new DNA molecule formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The chromosome which had undergone duplication during S phase of interphase now begins to move towards opposite poles of the cell. The completion of prophase can thus be marked by the following characteristic events. First, chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Second, chromosome which had undergone duplication during interphase begins to move towards opposite poles of the cell. Each chromosome radiates out microtubules called esters. The two esters together with spindle fibers form mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleus, and the nuclear envelope. The metaphase. The complete disintegration of nuclear envelope marks the start of the second phase of the mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosomes is easily studied. At this stage, metaphase chromosomes is made up of two sister chromatids which are held together by the centromere. Small disc-shaped structures at the surface of the centromeres are called as kinetochores. These structures serve as the sites of attachment of spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by the chromosomes coming at the equator, which one chromatid of each chromosome connected by its kinetochore to spindle fiber form the one pole and its sister chromatid connected by the kinetochore to spindle fibers form the opposite pole. The plane of alignment of chromosomes at metaphase is referred to as metaphase plate. The key features of metaphase are: first, spindle fibers attached to kinetochores of chromosomes. Second, chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. Anaphase. All the onset of anaphase, each of iso uh, chromosome arranged at the metaphase plate is split simultaneously and the two daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole, and hence the leading edge with the arms of the chromosome trailing behind. Thus, anaphase stage is characterized by the following key events. First, centromeres split and chromatids separate. Second, chromatids move to opposite poles. The telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. This is the stage which shows the following key events. First, chromosomes cluster at opposite spindle poles and their identity is lost as discrete elements. Second, nuclear envelope develops around the chromosome clusters at each pole, forming two daughter nuclei. Third, nucleus Golgi complex and ER reform. Cytokinesis. Cytokinesis accomplishes not only the segregation, duplication of chromosomes into daughter nuclei or karyokinesis, but the cell itself is divided into two daughter cells by separation of cytoplasm, called cytokinesis, at the end of which cell division gets completed. In the animal cells, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant, cell wall formation starts in the center of the cell and grows outwards to meet the existing lateral walls. The formation of the new cell wall begins with the formation of the simple precursor called cell plate that represents the middle lamellae between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of synis- synistium. This is the entire presentation. I hope I have presented well. Thank you.